We're official.
Okay, well, I don't want to take up too much of Miriam's time, so I'll introduce you now, and as a few more people are joining us. So we have two great student talks today. The first one will be from Miriam Bell in mechanical and aerospace engineering. Miriam works with Professor Rangamani and Professor Jean Zhang in pharmacology, and we also have another one of her committee members here, Dr. Bartol. So she is going to be talking to us today on her research on crosstalk between signaling and trafficking of AMPA during synaptic plasticity. Miriam?
Awesome, thanks. I will get set up. Okay, hopefully you can all see my screen.
Yep.
Good, perfect. Okay, so thank you so much for the introduction. So as Professor McCulloch already said, today I'll be discussing a computational model of synaptic plasticity in dendritic spines, specifically in regards to the receptor AMPAR. Okay, so to begin with, we're talking about neurons. And so just as some background, neurons communicate at synapses, as you see in this picture on the left here, and they must be able to modify the strength of their connections in order to adapt and have proper neural function. So synaptic plasticity refers to this ability, the ability to change the synaptic connection strength between neurons, and it can involve both biochemical and structural changes. So as seen in the schematic, there's a presynapse and a postsynapse, and for this talk, we'll be focusing on the postsynaptic neuron that receives the signal from the presynaptic neuron. And specifically, we'll be discussing dendritic spines on the postsynaptic neuron and how they change and adapt. So to talk about that, we need to know what are dendritic spines. So the majority of excitatory synapses are housed in dendritic spines, which are small protrusions along the dendrites of neurons. And they're actin-rich, and they compartmentalize biochemical, electrical, and mechanical responses. They also have characteristic shapes that are associated with their function, although this shape-function relationship is not fully understood. So here are some of the characteristic shapes. You have filopodia, thin, stubby, mushroom, and cup-shaped. Filopodia tend to be most common in neurodevelopment, while for mature spines, we typically talk about thin and mushroom-shaped spines. So we also know that spine shape and density can change over time. So this is a plot just showing how spine density changes during neural development, and then also typical spine shape and densities for different disease states. So we do know that spine shape is somehow correlated to function, but we're not entirely sure how. So during synaptic plasticity, dendritic spines undergo biochemical and structural changes, and that's what we're going to be discussing today. Okay, so AMPAR has emerged as a readout for synaptic plasticity because its density at the synapse changes during long-term potentiation and long-term depression. So just to take a step back, AMPAR is a glutamatergic receptor that binds glutamate and lets ions in to depolarize the membrane at the synapse. So as you see in the schematic, long-term potentiation involves an increase in AMPAR, which makes the synapse more sensitive, and it also involves an increase in spine volume. Long-term depression involves a decrease in AMPAR and a decrease in spine volume. So previously, it was thought that synaptic plasticity could be correlated to upstream signaling dynamics, such as that of calcium, or in particular, CAMK2. And much of our understanding of this correlation comes from really beautiful modeling papers done by people such as John Lisman. However, more recently, experimental evidence has presented a potentially more complex picture of this relationship. So CAMK2 is an abundant protein in neurons, and we do know that its dynamics are associated with synaptic plasticity, but we're not entirely sure how. So we were intrigued by the relationship between CAMK2 and AMPAR, and we wanted to understand how CAMK2 dynamics could influence AMPAR dynamics, specifically at the postsynaptic density, or PSD, at the top of the spine head. So said another way, we wanted to understand if we could predict AMPAR dynamics by looking at CAMK2, since CAMK2 is upstream signaling-wise of AMPAR. Additionally, we wanted to know what factors influence that signaling interaction. So these questions were driven in part by several open questions in the field regarding both CAMK2 and AMPAR. So regarding CAMK2, while it's been extensively studied, there's recently been some debate over its exact dynamics. In particular, it's known to autophosphorylate, which can drive an elevated sustained concentration of active CAMK2, but more recent experimental evidence has shown transient CAMK2 dynamics. And then regarding AMPAR, it's known that the density of AMPAR at the synapse changes during synaptic plasticity, but what trafficking mechanisms are responsible for that change is less understood. And currently there's debate over the relative contributions of membrane diffusion, endo and exocytosis, and the role of extrasynaptic pools of AMPAR, as you can see in this schematic here. Okay, so many of the trafficking modalities are inherently spatial. You need to have space to consider membrane diffusion, for example. So it's ultimately beneficial to consider spine morphology in all these questions as well. As I mentioned before, spines have characteristic shapes and they change size and morphology in response to activity and during synaptic plasticity. Therefore, we want to ultimately build models to address this coupling. So the coupling between signaling, AMPAR dynamics, and spine morphology. So we sought to address the overall question of coupling between signaling and morphology by tackling the open questions that I just discussed. And I'm going to give you the two takeaways of this talk first to really try to drive them home. So the first takeaway is regarding the consequences of different CAMK2 signaling networks that could underlie synaptic plasticity. So we found that network architecture could filter upstream signaling and trafficking effects, including that of model stimulus and initial condition effects. And second, we investigated how the morphology of dendritic spines influences signaling, and we found that spine morphology showed volume-dependent sensitivity to changes in bound AMPA steady state, which held across both idealized and realistic spine morphologies. Okay, so today I'm going to be focusing on this first question of biochemical complexity and trafficking. So there are various signaling species involved in the signaling cascade that leads to changes in AMPAR at the PSD. And these species span a fairly wide range of timescales, as do the different trafficking effects, as you can see here. So the question we wanted to address was, how does the choice of CAMK2 model influence the potential signaling propagation and signal integration across these different timescales? So to answer this question, we built a computational model of deterministic differential equations that describe the interactions of key signaling species known to be involved in synaptic plasticity. We constructed two different signaling networks for CAMK2 and phosphatase dynamics. First model, seen on the left here, is called bistable, and it reproduces CAMK2 dynamics that could produce a sustained elevated steady state for CAMK2. So this is referred to as bistable because depending on initial conditions or stimulus input, CAMK2 can have two different steady state behaviors, one elevated and one zero. The second model is called the monostable model, and it reproduces transient CAMK2 dynamics. And it's called monostable because it has a single steady state that is always zero. Okay, so to address the second open question on AMPAR trafficking, we included two different trafficking modalities here. The first is endo and exocytosis. Endocytosis we determined to be PP1 dependent, and exocytosis is CAMK2 dependent. And then we also included a CAMK2 mediated influx of membrane AMPA. So this is to represent that extrasynaptic pool of AMPA that you can have diffuse into the spine. And diffuse here just means that it's a biochemical influx. Okay, because we're focusing on biochemical complexity here, we're using a compartmental model, and therefore we don't have spatial complexity and can't consider membrane diffusion. So the last step of this model describes free AMPA binding to PSD95, which is a scaffolding protein, to produce this complex bound AMPA. And this is going to be our ultimate readout for this model. Okay, so the vast majority of these equations were described through mass action kinetics or Hill functions. And the goal here is to consider the key species across multiple timescales and how the signal can propagate through each of these respective model setups. We ran these two models through various different biochemical and trafficking variations, but due to time constraints, we'll only be considering a subset of these. So to ground everyone with the difference between these two models, I'm going to highlight the steady state behavior of CAMK2 and how it translates to bound AMPA dynamics. So the top row of panels here is the steady state for active CAMK2. This is the steady state for bound AMPA. The left column is bistable, right column is monostable model. So we're plotting these different steady states for different CAMK2 initial conditions on the x-axis and for two different concentrations for PP1. Low concentration of PP1 in blue and high concentration of PP1 in red. Okay, so if you look at the left column, the bistable model can have two different steady state behaviors, high or low, within a certain initial condition regime, as you see here, and is dependent on the PP1 initial conditions. So there's a coupling between CAMK2 and PP1 that determines the steady state behavior for CAMK2, and that translates to bound AMPA levels that also have this coupled dependence between PP1 and CAMK2. In comparison, the monostable model always has a zero steady state for CAMK2, and it only shows dependence on CAMK2 initial conditions for bound AMPA. Okay, so with that understanding, we're now going to consider different model stimulus. So previous work has indicated that CAMK2 can act as a leaky integrator of calmodulin input. So calmodulin is a species that's upstream of CAMK2. So we wanted to investigate if either of our models showed sensitivity to active calmodulin pulses, shown here at 3 different frequencies and 2 different magnitudes. So I haven't shown this here, but for the bistable model, we found that due to autophosphorylation and rapid CAMK2 activation, CAMK2 and subsequently bound AMPA showed no dependence on input frequency. In contrast, the monostable model did exhibit leaky integrator dynamics. So these are the temporal dynamics for active CAMK2 for 3 different frequencies and 2 different initial conditions for CAMK2 for the smaller stimulus I showed here. So we wanted to understand how this CAMK2 temporal dynamics translates to bound AMPA. So this plot is showing the steady state of bound AMPA for these different conditions. And we see that the system is both sensitive to stimulus magnitude and stimulus frequency. And you can actually display different trends within those factors. Therefore, the big conclusion from this is that model architecture determines the influence of upstream signaling, and post facto, this conclusion may seem pretty obvious. But however, due to the inherent nonlinearities in the bistable model and numerous species interactions in the monostable model, it's not actually obvious just looking at the model architecture that these effects would be present. Additionally, these 2 models displayed their nuances differently, either through steady state behavior or temporal dynamics. And so modeling allowed us to, parse these out and examine these differences. Okay, so up until now, we've considered the biochemistry of the system, but what about trafficking? So we know that AMPA must be physically moved to the PSD. So how does our model architecture influence trafficking modalities? So to investigate this, I would be really clear about the different trafficking forms here. Specifically, we have endo and exocytosis, which you can see the equation for here. And then we also have this CAMK2 mediated influx of AMPAR. And when we have influx into the system, we don't actually have mass balance because we have this external source of AMPAR coming in. If we have no influx, then we have mass balance for the system. Okay, so if we look more closely at these endo and exocytosis terms, you can see that we have a species-dependent term, this C1 and C2, and then we have a basal term, C3 and C4. So we want to consider the system with and without an influx term that I don't show. And then we want to scale the relative contributions of endo and exocytosis in 2 different ways. The first way is by modifying C1 and C2, so activity-dependent term. And the second way is by scaling the entire contribution of endocytosis or the entire contribution of exocytosis. Sorry, I'll repeat that. So we're going to change this in 2 different ways, either scaling C1 and C2, and the second way is scaling the entire terms. Okay. So this result might look a little intimidating, but we're going to break it down panel by panel. So if we consider a system that only has endo and exocytosis, so there's no influx, we're looking at the top 2 panels here. If we scale the activity-dependent term in A, and consider both models here and two different concentrations for CAMK2 initial condition to get high and low CAMK2 steady state for the bistable dynamics. We see that we can achieve new bound AMPA steady state values, but only for the bistable case. And it's a little hard to parse out all these different colors, but just take my word on it. You only achieve new steady state for the bistable model. If we scale the entire term, to make sure that we're at steady state at the beginning, we run our system to steady state without a stimulus and then apply the stimulus at 500 seconds, as you see here. And we see that without an influx, we don't change from steady state. And we do, however, have a condition here where before we have our stimulus, it actually drives bound AMPA to 0, and you remain at 0 from there. Okay, so considering the system with influx, this C and D panels, we see that we always achieve a higher steady state for bound AMPA when scaling C1 and C2. So it's always greater than 1,000. And for the whole term, this panel D, you also always achieve a higher steady state other than this one case down here where endocytosis is maxed out. Okay. So, sorry. Here's the slide I want. Therefore, in order to achieve a new steady state for AMPAR, we need one of two different ways to achieve this, essentially. You either need a constant forcing term, say elevated CAMK2 or PP1 in the bistable model, or you need an external influx of AMPAR. So the bistable model has a constant forcing term. So even without influx, it can achieve a new steady state, as you can see here in panel A. And it's actually interesting to note here that you can achieve long-term potentiation, which is an increase in AMPAR, or you can also achieve long-term depression, which is a decrease in AMPAR. However, for the monostable model, it doesn't have a constant forcing term because it has transient dynamics for CAMK2. So it can only change its steady state behavior through an influx of membrane AMPAR. So that's panels C and D. Thus, we concluded from this that trafficking effects are actually dependent on underlying biochemical signaling networks. All of these results have been for a compartmental system of ordinary differential equations. However, we want to consider the role of geometry on these biochemical effects. So to do that, we translated our compartmental model into a spatial reaction diffusion model with the proper boundary conditions. Due to time constraints, I'm not going to walk through this model development. However, we've done this previously with other biochemical signaling models. And this model setup allows us to systematically consider the role of geometry and full spatial trafficking effects. So here we specifically considered idealized thin and mushroom spines of different volumes. And just as some background, thin spines tend to be smaller and more dynamic compared to mushroom spines that tend to be larger and more stable. Additionally, we considered reconstructed realistic spines from 3D electron microscopy images here on the right. And it should be noted that obtaining EM images and meshing them to sufficient quality to run finite element methods simulations on them is non-trivial. So these results are actually like 5 years in the making. So took a long time. Okay, so here we scaled thin and mushroom spines to different volumes from their control volume. And we're looking here at % change in bound AMPA steady state from its initial steady state baseline. So you can see here in both models, you see this clear resetting of bound AMPA change between the two shapes. So this is the thin spines in blue and the mushroom spines in red. Additionally, we get a much steeper increase in % change for the smaller thin spines compared to the larger mushroom spines. And if you compare between the models, you see this similar reset between spine shape and size, but for the monostable model in the here for the mushroom spines, you actually get this non-monotonic trend for the mushroom spines. However, it's important to note that the % change, like actual value for the monostable model, is much lower than the change for the bistable model. So these findings support the idea of smaller thin spines being more biochemically dynamic compared to these larger mushroom spines. Okay, so we wanted to see if these trends held for realistic geometries. So here I'm plotting the same figure as before for the bistable model with thin and mushroom spines, but I'm now showing the results for simulations in 7 realistic spines of different morphologies. So you can see here that While the trends aren't as clear as the idealized case, the realistic spines shown in yellow still show higher percent change for smaller morphologies, with this transition region around here where the percent change seems to reset. Okay, so to summarize, we looked at two potential signaling networks and found that model architecture could filter upstream signaling and trafficking effects, including stimulus and initial conditions. Using a spatial model, we found that spine morphology showed volume-dependent sensitivity to change in bound and to steady state, and that held for both idealized and realistic spine morphologies. I should say that the work here is in review and in preparation, but we're just brushing the surface on many open questions with this work. For instance, here we're focusing on how morphology influences signaling, but we need to complete this feedback loop and consider how signaling can influence morphology, specifically during synaptic plasticity, where you do see these changes in morphology over quite short timescales. Additionally, we want to more closely consider how these different biochemical and trafficking modalities could represent disease states to better understand their implications. And with that, I'd like to acknowledge my co-authors on this work, specifically Dr. Lee and Professor Rangabani, my lab and funding sources for their support, and all of you for your attention. And I can take any questions. Thank you.
Thank you, Miriam. As you can probably tell, Miriam's done a lot of work and she's pretty much ready to defend her thesis soon. So questions for Miriam?
Can I go ahead and ask you a question?
Yeah.
Yeah, that was really cool, Miriam. So I was curious, when you're looking at the more realistic spine geometries, there were a handful that were really close to the thin kind of models that you had. And so if you look at those visually, are they pretty much like a thin dendrite?
Yeah, so you do see this variety. So it is quite interesting because part of the, I haven't gone to do as much analysis on these realistic spine morphologies as I would have liked to do. That's still a work in progress. But you do see that like these ones tend to be, all of these shapes you would classify as thin, but they do come in quite different volumes. And so we're trying to figure out if it's a volume to surface area ratio that matters, or if it's volume, or if it's like some ratio of, PSD area that also can change between these different morphologies. So we're not entirely sure, but it does seem to like, you do get a pretty nice trend. That being said, I'm very happy you asked this question because I included this figure. So we considered, this is plotting that same result here versus volume, but if you consider the same results versus volume to surface area ratio, there's actually 3 spines of different volumes that share the same volume to surface area ratio, which is all here.
Oh, that's super interesting.
And so we're not entirely sure what geometric factor basically we should be plotting against. That's something we don't know. So is it volume that matters? Is it volume to surface area? We don't really know.
Yeah, that's cool. That's super interesting. Yeah, and I'm sure there's a million things you could look at there. But anyways, that was awesome. Thanks so much.
Any other questions for Miriam? Well, if not, I would like to wish you very well in your defense, Miriam. I know it's going to go very well. I also want to thank you for being such a enthusiastic and active participant in the Interfaces program during your PhD, and not to mention a great TA, and now also in the past couple of years an instructor for one of our courses. So I really want to thank you and wish you well.
Thank you. I've had a really great time as part of the Interfaces program, so I appreciate it.
So next up we have Abby Teitgen. Abby is working on multi-scale modeling of cardiac muscle to understand the effects of changing ATP to deoxy-ATP with the ribonucleotide reductase. It's involved in a collaboration with experimental group in the University of Washington of Mike Rainier. And she's really working towards maybe the first computational model that will start at the atomic resolution and end at whole organ scale. So with that, I'll pass it. I don't want to look down and read the screen of my phone, so I can't remember what the name of the title of her talk is.
Awesome. Yeah, thank you so much for the introduction. Okay, can everyone see my screen? Cool. So yeah, for those of you who I haven't met, I'm Abby. I'm a fourth year PhD student in Dr. McCulloch's lab, and my interdisciplinary co-mentor for interfaces is Dr. Jeff Omans. I'm interested in studying heart failure. So as I'm sure we're all probably aware, heart failure is a major cause of morbidity and mortality. It affects around 40 million people worldwide, and it's the leading cause of death in the world. So the term heart failure is kind of an umbrella term that refers to a number of different syndromes that have to do with impairments in cardiac function. So the two kind of major classes that we often talk about are heart failure with reduced ejection fraction and heart failure with preserved ejection fraction. And we can also think about heart failure in terms of the different things that can cause it. So for example, myocardial infarction is a major cause of heart failure, and there can also be other genetic and environmental causes as well. So I specifically am interested in dilated cardiomyopathy. And so this involves a dilation of the ventricles and a thinning of the walls, and the heart is no longer able to pump enough blood to meet the demands of the body. And in general with heart failure, we also see energetic impairment. So the heart is generally in a state of energy starvation where the mitochondria are not able to produce enough ATP. So for those of you who don't study the heart, the fundamental contractile unit of the heart is the sarcomere, and this contains thick and thin filaments. So the thick filaments here are shown in pink, and the thin filaments are in blue. So the thick filaments are myosin, and the thin filaments are predominantly actin. And we also have a number of different regulatory proteins on the thin filament. So we have troponin here, and then we have tropomyosin, which is the purple protein. So generally what happens during muscle contraction is that calcium binds to troponin, which then moves tropomyosin out of the way of the actin, or of the myosin binding site on actin. So myosin can then bind and pull on actin to shorten the sarcomere and generate force. And so the overall process here is referred to as excitation contraction coupling. So we first have our electrical stimulus, which is the action potential, and then we get an increase in calcium, which leads to an increase in force. And then as calcium decreases, we get relaxation. So because of advancements in genetic screening, we now have a much better understanding in the genetic causes of heart failure. So here you can see the most common mutations that lead to dilated cardiomyopathy, and a lot of them are in the sarcomere, as you might expect. And so when we think about ways to treat heart failure, one strategy is to try to directly target the sarcomere to fix the underlying mechanical dysfunction. And so that's kind of where my research comes in. And it's also important to mention that although we do have a much better understanding of these underlying genetic causes, the link between genotype and phenotype is not always very clear. So that's where computational modeling approaches can be really valuable in linking these really small scale effects on sarcomeric proteins, and then how does that scale all the way to changes in ventricular function. So I'm interested in studying a small molecule called 2-deoxy-ATP, or DATP, and it's a naturally occurring nucleotide, very similar to ATP, but it's missing a hydroxyl group. And this is being investigated as a heart failure therapeutic. So DATP has been shown to directly act on myosin to increase steady state force and the rate of force redevelopment. And it's also been shown to increase cell shortening and force production. In addition to its effects on myosin, it also has been shown to increase the rate of calcium transient decay, which results in faster relaxation. And it has also been shown to improve ventricular function in a number of animal models. And we also have seen that levels of DATP as low as 1 to 2% of the overall nucleotide pool significantly improve contraction, but the mechanisms behind this are not very well understood. So my goal for this project is to develop a multi-scale modeling framework extending from the molecular level all the way up to the full organ level to assess how changes in myosin with DATP scale to changes in ventricular function, both in a healthy heart and in the case of heart failure. So we started with molecular and Brownian dynamic simulations of myosin binding to actin with DATP. And then we used this to constrain a Markov model of sarcomere mechanics, which is also coupled to a myocyte energetics and calcium dynamics model. And then this is embedded within a biventricular mechanics and lung parameter circulation model. So the Sarcomere model that I've been working with for this study was published in 2020, and it contains 6 states. So the first state is super relaxed. So this is a state where the myosin heads are essentially off. So they cannot participate in cross-bridge cycling or generate force at all. And the super relaxed state is actually still an ongoing area of research. And we don't fully understand exactly what's happening there. The next state is N, which is non-permissive. So here there's no calcium in the system. Myosin is not bound to actin. Next, we have the permissive state. So we have calcium bound to troponin, but myosin still has not bound to actin. And here we have ADP bound to the myosin. So this entire process of cross-bridge cycling is driven by the process of ATP hydrolysis. Next, we have state A1. So this is loosely bound. And then state A2 is strongly bound. So myosin is bound to actin in both of these states. And then state A3 is post-power stroke. So going from A2 to A3 is the actual power stroke where myosin is pulling out actin to generate force. And so we also have several viscoelastic components to the model, and this allows us to simulate whole cell mechanics as well as scale up all the way to the full ventricle. So the first thing that we wanted to look at was whether having DADP bound to myosin results in a difference in association rate to actin compared with ADP bound myosin. So to do this, we used Brownian dynamics simulations. This is done by Marcus Hoek, and you heard him present on this a couple of weeks ago. So in Brownian dynamics, essentially, we're treating the molecules as rigid bodies, and we run thousands and thousands of trajectories and allow them to move around, and they either bind or they don't bind. And we can use that to get an association rate. So this is a plot of association rate versus reaction distance. So reaction distance is a parameter in the model that basically defines the distance at which the two molecules bind. And so rather than choose an arbitrary reaction distance, we ran our simulations for a range of reaction distances. And you can see that no matter what reaction distance we choose, we get a higher association rate with DADP compared to ADP. And if we specifically look at the reaction distance that corresponds to the ADP association rate experimentally measured from the literature, this corresponds to around a 2.7-fold increase in association rate for DADP. So the next thing that we wanted to look at was how does this affect sarcomere mechanics? So in our model, the myosin-actin association rate corresponds to this parameter Ka. So that's what we're looking at here. And so the first thing that we did was to optimize our model to match experimental data. So what you're looking at here is called a forced PCA curve. And what you do is essentially you slowly increase the concentration of calcium in the muscle, and then you see how much steady state force it can produce. And PCA is the log of the calcium concentration. So a larger PCA is a smaller calcium concentration. And the forced PCA curve has this kind of S shape to it because of a number of factors related to cooperativity in the sarcomere. So this has to do with things like overlap of neighboring tropomyosin molecules. So if one tryptomyosin moves out of the way, the one next to it will get pushed out of the way as well. Or if you have one myosin head bound, it will kind of pull the one next to it down as well. So that contributes to this kind of S-shaped curve here. So the open circles are experimental data for ATP and DATP force PCA curves. And then the solid black line is our model prediction. So this was, again, optimized to match the wild type data. And then when we increase Ka to based on the Brownian dynamics data, we get this blue curve. So you can see that it shifts to the left. So we have increasing calcium sensitivity, meaning that we get more force at the same level of calcium. And overall, we get an increase in steady state force, but not quite to the extent that we saw experimentally. So the next thing we wanted to look at was whether there were additional parameters in our model that we could adjust to better match the data for the DATP case. And we found that if we also increased these parameters KD, K1, and K3, we see a much better match to the experimental data. So this suggests that in addition to increasing the association rate between actin and myosin, DATP is also potentially increasing additional rates in the cross-bridge cycle and increasing the overall rate of cross-bridge cycling. And this matches well with previous modeling results from Merck as well. And so using these same parameter adjustments, we were also able to reproduce slack restretch data. And so in a slack restretch protocol, you basically allow the muscle to contract isometrically at an initial length, and then you shorten it really quickly to break the attached cross bridges, and then you stretch it back out to the original length and let it redevelop force. And so the combination of these force PCA and slack restretch experiments tells us a lot about the underlying kinetics. And so we're basically able to reproduce both of these using our model for DATP by making these 4 parameter changes. So the next thing we wanted to look at was cell shortening with DATP. So when we make these 4 parameter changes that I just showed, what we noticed is that when we looked at cell shortening for varying percentages of DATP, we didn't see much of a change in shortening for smaller amounts of DATP. And so what you're looking at here is relative cell shortening. So more shortening means more force, basically. And so if we compare the 2% DATP case to ATP, there's not much of a change. But experimentally, we've seen that we do get significant increases in shortening with lower amounts of DATP. So we found that in our model, if we also increase this parameter K force, which has to do with the transition of myosin out of the super relaxed state, we see a much larger increase in shortening for only 2% DATP. And this matches with what has been shown experimentally. So this would suggest that DATP is also increasing the transition of myosin out of the super relaxed state. So it's basically resulting in more myosin heads available for cross-bridge cycling, in addition to its effects on the actual cross-bridge cycle. So at this point, we were able to reproduce the force PCA and slack restretch data, as well as shortening data. But when we looked at relaxation, we were seeing slowed relaxation with DATP, whereas experimentally, DATP has been shown to increase relaxation. So we next wanted to see whether incorporating the effects of DATP on the calcium transient could explain this enhanced relaxation. So here you can see model-generated twitches from the model. This is now at the tissue level. And so we're starting with ATP in black and then scaling all the way up to 100% DATP in blue. So for this simulation, this is just adjusting these 4 cross-bridge cycling parameters, Ka, Kd, K1, and K3. And again, here we see a linear relationship between force and DATP percentage, and we don't see much of an increase in force for lower percentages of DATP. For this simulation, we are just scaling the transition out of the super relaxed state with DATP. And here again, we see much larger increases in force with only 2% DATP, but still we see slowed relaxation. So here you can see the effects of DATP on the calcium transient. So we get a faster relaxation of the calcium transient. And then when we incorporate that into the model, we get faster relaxation of the twitch, but also lower peak force. And so finally, if we combine all of these effects together, we get a significant increase in force for 2% DATP. And we also see faster relaxation. So essentially, we need to account for all three of these factors together to fully explain what we've seen experimentally with DATP. And then the last thing that we wanted to look at was how the effects of cooperativity interact with each of these three effects. So for this simulation, we set the cooperative activation parameter in the model to be 0. And so when we lose cooperativity here, we see a much more blunted effect with DATP. And so this would suggest that nearest neighbor cooperativity is also something that could be important in terms of underlying mechanisms. So we finally wanted to use our model to predict the effects of varying percentages of DATP on ventricular function. And so here you can see pressure volume loops simulated in our model. The dashed black line is the healthy wild type simulation. And then we are applying the same increasing percentages of DATP here. And then over here, I have more of a quantification of some different metrics of ventricular function, things like ejection fraction, cardiac output, fractional shortening. These are all things that we look at to see how well the heart is performing. And in general, we see dose-dependent improvements in all of these metrics. We also saw changes in the plus and minus dp/dt. So these are the rates of pressure increase and decline. And so this means that DATP is also increasing relaxation at this level as well. So we also wanted to see what would happen in the context of heart failure. So the heart failure simulation in our model is shown in the green dashed line. And then again, we applied the same percentages of DATP. So in the failure case, we see a decrease in fractional shortening, ejection fraction, and cardiac output. And then when we apply increasing amounts of DATP, we see, again, dose-dependent improvements in function. So the last thing that we wanted to look at were the effects of DATP on energetics. So like I mentioned before, in heart failure, we have changes in energetics that can occur. And so we wanted to see whether DATP would lead to further impairment in energetics or an improvement. So this is for the healthy case. Again, this is for the same percentages of DATP. And so I have on the top concentrations of ATP, ADP in inorganic phosphate. And then I have some other metrics of energetic function here, like myocardial oxygen consumption, creatine phosphate ATP ratio, which is a good indicator of metabolic function, and then cross-bridge cycling and ATPase rate. So in general, with DATP, we see increases in ADP and inorganic phosphate concentrations at higher levels of DATP, and a decrease in the creatine phosphate ATP ratio. And so this matches up with what has been measured experimentally with DATP. In the failure case, we see elevated inorganic phosphate and a decrease in the creatine phosphate ATP ratio. So this is pretty indicative of heart failure. And then we see similar trends when we apply DATP in terms of increasing inorganic phosphate and ADP concentrations. We do see a slight increase in myocardial oxygen consumption with DATP, which is potentially a concern with DATP treatment, but it does not appear to increase beyond the healthy level. And we also see increases in efficiency, both in the healthy and in the failure case with DATP. And interestingly, efficiency also increased to a greater extent in the failure case. So in conclusion, we have developed this novel multi-scale modeling approach, which allows us to link molecular scale changes to shifts in ventricular function. Our model predictions indicate that elevated DATP leads to a synergistic effect between increased cross-bridge binding and cycling, increased superrelaxed state recruitment, and increased calcium transient decay, which results in increased contractility and lusitropy. We also saw that elevated DATP improves ventricular function in a dose-dependent manner in both healthy and failing conditions without further impairment of metabolic function. So in terms of next steps, there is a number of things that I think would be really interesting to do. One thing would be to extend this to a finite element framework. So we could look at things like regional differences in mechanics. And we also have some data on using DATP in a myocardial infarction model. So that could be interesting. Another thing that I am interested in doing is adding a growth model to assess the impacts on ventricular function over time, because with heart failure, you're going to have changes over time in terms of ventricular hypertrophy. And so it would be interesting to look at how DATP affects the heart at different stages. There is also definitely a lot more work that needs to be done in assessing the effects of DATP on the super relaxed state, and whether or not cooperativity is important in this as well. So we could combine experimental and modeling approaches to look into that further. And finally, this model could also be extended to assess additional small molecule therapeutics beyond just DATP, or looking at sarcomere mutations, like I mentioned at the beginning. So with that, I would like to thank Dr. Andrew McCulloch, as well as the rest of the Cardiac Mechanics Research Group and our collaborators at the University of Michigan, the University of Washington, and Simula Research Laboratory in Norway. And I'd be happy to take any questions.
Thank you, Abby. We have a very clear presentation. We have time for questions for Abby.
So I have a question. It might be maybe a little bit out of bounds, maybe, but question is, wouldn't DATP be a great drug if you were an athlete?
So you mean like to further improve cardiac performance in a healthy heart?
Yes.
Yeah, that's an interesting question. I think that's one of the things that we wanted to kind of get at with the model is there a point at which you would start to see negative effects from too much DATP? And I think that could potentially happen in the healthy heart, but I don't really have any data on that.
Yeah, I mean, also skeletal muscle is affected too, although it doesn't seem to affect fatigue resistance. So it affects skeletal muscle force development, but doesn't make it more fatigue resistant.
Well, there's probably another drug that'll do that.
Yeah. Just come up with a new illegal substance.
Yeah. Well, after the Olympics, you know, it's been on my mind a little bit.
Yeah, definitely.
Yeah,
Ivan?
I've got a quick question. Is the super relaxed state associated with like cardiac pathology? And is that like does that play into conserving ATP? I recall you mentioning that earlier on, that like kind of a failing heart is having a hard time acquiring ATP. Not sure. Yeah, sorry if that's a jumbled question.
No, yeah, that makes sense. Yeah, I think potentially, I mean, I know it definitely has been associated with different drugs beyond just DATP. And I think it is also associated with heart failure, but there's still a lot about the super relaxed state that we don't really know.
Abby, can I jump in for a second?
Yeah.
The other thing I know, kind of the opposite direction, is that the super relaxed state has been associated with hypertrophic cardiomyopathies, so like overcontractile function, and so kind of the opposite consensus, but still leading to greater contraction. So it definitely is involved in different diseases.
Yeah, that's a good point.
And the hypertrophic cardiomyopathies are often associated with some sort of gain of function. So the similar sort of effect that deoxy-ATP could have in making the super relaxed state less stable, and therefore increasing the likelihood of cross-bridge cycling. You know, if you have a mutation which does the same thing, then would be a gain of function mutation that could lead to hypertrophic cardiomyopathy. Any other questions for Abby? Well, if not, I'd like to thank again both Abby and Miriam for really well prepared and nice talks, everyone for your participation, Dr. Bartol for coming along, Kelly for organizing, and well, wishing you all a good week.
Everybody remember to RSVP for the symposium. We want to see you there and feed you lunch.
That's right.
Thank you, everyone.
Thank you.
Thank you.
Hi, thank you.